Annapurna, so we'll start after 5 minutes.
Sure. Should I share my screen by then?
You can do that, yeah.
Is it visible?
Yeah.
Okay. Hi, Pranjal. Mm, sure. Mm. Yeah, it's been open all through, as in the institute itself. Yeah, I mean, the lab work was obviously affected for a few months because of. Really? When was this? Yeah, yeah, it's one of the good canteens that you can find in institutes. Yes. When was this though? Yeah. Oh yes, yes. Oh yes, yes. When were you here though? Oh, oh, did you come for that observership program? Ah, okay, okay. Yes, yes, yes. Hmm. Yes. Yeah, I will convey the message to everyone that our observership students still remember us. Yeah, I'm super surprised that so many people have actually joined considering today's Dashera. And yeah, I noticed. I saw Shayantan's talk on YouTube later, and there pretty good participation. Yes.
People are still joining in though. It became double number in like 2 minutes.
Right, right. Mm, I saw. Yes, yes. Mm-hmm. Mm-hmm. Nice. Mm-hmm. Yeah, I can imagine. Like in CCM, we've been like a conference is under work, something that's called High Sci or Hyderabad Science. So yeah, that's going to be in December. So yes, I get
Is
it.
is it is it still happening?
Sorry.
I mean, this December it would be happening.
Yes, yes.
I mean, offline or
Online,
somewhere
of
else.
course.
Okay, cool.
Online. Yes.
I do like the idea of online conferences, though it's like very
Mhm.
accessible to people.
It is. So while we are on conferences, since you people are organizing Neuro November, I just saw this tweet by Bias Watch. It's a
Yeah,
I don't know if any of you saw that. So they are asking for applications from conference organizers. If you have a good gender equity in the number of in your speakers, at least or more than 30% of non-male speakers, and in addition, if you have a discussion on gender equity in science, they are ready to give you certain monetary help with the conference organization. So
we we have 1:1 probably.
great. So you people might consider applying to that. I'll send the link to one of you people.
We is that on Twitter?
Mm. It's on Twitter.
Yeah, so we
Yes.
already got a lot of leads. Even Vidita
Mm.
probably retweeted.
Yeah, yeah, yeah. She must have. Yes. Yes. Yeah.
So it's 36 now. Should we start
Sure.
or should we give 5 more minutes? What do you think? Perfect. So I'll start with a brief introduction. Just give me a second. So, okay. No. So we have Annapoorna with us today, and she'll be presenting on stress, brain, and mental illness spectrum. She is a PhD candidate working with Dr. Arvind Kumar in CSIR CCMB Hyderabad. She studies the function of histone lysine demethylases in mediating the effects of stress on brain, with special focus on adult neurogenesis. Along with neuroscience, she is passionate about science communication and outreach. I th- she's been involved in outreach related programs in CMB, including organizing what she just mentioned, HiSci, that's a conference for people who love science. so she's also written articles for India Bioscience and the Quid. In addition to communicating science, she strives to normalize discussion on mental health and mental illness through her writing. So today's talk would be centered on two themes. One part related to her PhD work, where she'll discuss the effect of stress on brain, focusing on epigenetics. The other part would be about the spectrum of mental illnesses and the upcoming idea that they cannot be categorized into silos, but actually lie on a spectrum. So over to you, Annapurna.
Thanks a lot, Ankush. Just let me know if my screen's visible.
Your screen's visible.
Great. Okay. Good evening, or whichever part of the day it is for people who are not joining from India. So it's great to have you all here. And as Ankush already mentioned, I'll be talking about stress, brain, and the mental illness spectrum. So, at the beginning, at the outset, I'd like to say that I am not going to discuss any data per se. I'm going to be talking about what I and others in my lab work on in general, just giving an idea of the techniques we use and why we do these studies. That will be one part of my talk, and the other part is going to be about the mental illness spectrum, the details of which I'll discuss further. Okay. So to start with the talking about stress and the brain, the first thing I like to talk about is the stress response. So this, I suppose, most of you would already be aware of. The primary stress response pathway in our body is the HPA axis, or that stands for the hypothalamus pituitary adrenal axis. So the way this works is whenever the body perceives stress, the hypothalamus in the brain releases a hormone called as the corticotropin releasing hormone or corticotropin releasing factor, which further stimulates the release of ACTH, that is the adrenocorticotropic hormone, which goes into the body's circulation, finally reaching the adrenal glands near the kidney and stimulating the release of cortisol. So I suppose everybody would have heard of cortisol because that is the hormone that is most well associated with stress. In fact, there are suggestions in the field that by measuring the level of cortisol in an individual's bloodstream, one can decide, one can understand what is their stress level. And here I'd like to mention that in rodents, there is a homologue of cortisol that's called corticosterone. So going ahead, if I use corticosterone, just understand that I'm talking about rodents, which is the animal model I work with. So this is how the normal stress response works. Cortisol is released, and once a certain level of cortisol has been reached in the blood, there is a feedback regulation that happens. So the cortisol goes back to the brain, to these regions, hypothalamus and the pituitary, which have receptors called glucocorticoid receptors on the cells. So cortisol binds to these receptors, ultimately leading to a suppression of the stress response. So that's how this cycle is maintained. And the suppression is also necessary because cortisol has a myriad of effects on the body, metabolic effects, effects on immunity, all of which are directed at handling the stress that you are facing. So once this stress period is gone, this response also needs to go. So hence this feedback is important. So what happens whenever there is a case of neuropsychiatric disease or disorder? Uh, these are characterized by a maladaptive stress response. So basically, the feedback that is supposed to happen that leads to a reduction in stress response, that does not happen. So how does it get manifested? So whenever we think of an individual in stress, these are the kind of things we visualize. Maybe the person is overdosing on coffee or any other addictive substance, or they're hyperventilating. So there are multiple ways stress gets manifested. Why does it get manifested in multiple ways? I'll talk about it further. But there is a good correlation between stress and susceptibility to developing neuropsychiatric diseases. In particular, among neuropsychiatric diseases, mood disorders. They're called mood disorders because there is a dysregulation of mood regulation. There's a dysregulation of mood in these diseases. And the three primary or well known mood disorders are depression, anxiety, and bipolar disorder. So, what leads to these diseases? There is definitely an angle of genetics involved. There are certain individuals who are genetically predisposed to develop these conditions. So there are several genes that have been associated, and most of these genes are those that are involved either in the regulation of the HPA axis, or they have some role to play in those brain regions that perceive stress and reward. But a major environmental risk factor that precipitates these disorders is psychological stress. So, but how does stress do this? Uh, so, initially, like in the initial years of when people, when the HP axis was just being understood, people realized that whenever there is extreme stress, there is a dysregulation in the HP axis, and which translates or manifests into behavior. But how is stress doing this? So, a very lucrative candidate for this is epigenetic mechanisms. So, epigenetics is anything that happens around the DNA rather than within the DNA. Changes within the DNA come under the purview of genetics. So, epigenetics is changes that happen around the DNA, but they ultimately affect the gene regulation or what comes out of the DNA. So, hence, they are super important in that sense. So, primarily, there are three very well known and well studied epigenetic regulatory mechanisms. The first that was discovered was DNA methylation. So when the chromosomes, which are made of DNA and histones, so within the DNA, the cytosine residue oftentimes is methylated, and this modification has implications for whether or not the DNA is further transcripted and later translated. Uh, Further, histones are the proteins around which DNA is bound. Histone tails also have several chemical modifications like methylation, acetylation, simylation, all of which also have implications for how the DNA bound around them gets, whether or not the transcription machinery can access the DNA around it. So there are several ways that this happens. Over time, the understanding of how these epigenetic mechanisms actually act has changed and evolved. Also, another well-known epigenetic mechanism is RNA interference or the effect of non-coding RNAs. So within non-coding RNAs, also there are several types. There are long non-coding RNAs. There are microRNAs. So one mechanism is RNA interference, where microRNAs or short RNAs uh, go and bind to your mRNAs and ultimately prevent their transcription. So, so these are the three well-known mechanisms, all of which have been associated with all of this have been which have been observed to be dysregulated in response to stress. Another interesting point that I'd like to mention here is something that's called as epitranscriptomic changes. So epitranscriptome is changes, chemical changes that happen on the RNA. So among these, RNA methylation is one such modification that is gaining a lot of interest in recent years. So uh, maybe once a few of you who are interested, we can discuss about that later. So once this idea that stress could be affecting the HPA axis and other brain regions through changes in epigenetic mechanisms, once this idea was proposed, several studies came about that have directly linked stress to these mechanisms, like acute stress affecting global DNA methylation patterns, stress leading to changes in histone modifying enzymes, uh, stress leading again to changes in histone modifications, and also in chromatin remodeling. So these are just a few examples, but there are hundreds of studies which came up. So, so this obviously excited neuroscientists beyond measure. That this is something that this aspect of understanding the brain's response or the body's response to stress was pretty much ignored before the past decade, and this poses a lot of importance, especially in the sense of translation, because epigenetic mechanisms were being already studied in the context of other diseases, especially in cancer biology, and in fact some chemical inhibitors and modulators which can affect these mechanisms were already being used in the clinic for as anti-cancer therapy. So it had it had an implication for possible drug development for neuropsychiatric diseases also. So talking further about the brain regions that are affected in mood disorders, because I spoke about the HPA axis, so which primarily is regulated by the hypothalamus and pituitary and a couple of other brain regions. But the range of symptoms that we see in mood disorders, it comes about because there are several brain regions that are involved in these functions of stress response and reward perception. So these are the major brain regions that are listed here: the prefrontal cortex, the nucleus accumbens, the ventral tegmental area, amygdala, and the hippocampus. So this brain image might look a little different from the images that most of you might have seen, because this is an outline of a rodent brain. Looks a little different than human brain. So among these brain regions, the VTA, nucleus accumbens, and the PFC, this particular pathway, this is one of the most important pathways that is involved in reward perception. In fact, all of these brain regions are involved in sensing or understanding reward and the feeling of pleasure. So, uh, if we look at the function of each of these brain regions, we can understand why the symptoms of each of the mood disorders come about. Like the VTA, NAC, and PFC are directly involved in reward perception. An issue or a dysregulation in these brain regions will directly affect reward sensing in us. uh, the hippocampus and the prefrontal cortex. The hippocampus is the seat of long term memory, and the prefrontal cortex is involved in deeper cognition. The hippocampus also has a role to play in cognition. So, an effect on these brain regions will ultimately affect memory and cognition. The amygdala is involved in fear response. So, an example would be people who have anxiety disorders have dysregulations in the amygdala and the hippocampus. So, among these brain regions, the one that I'm most interested in is the hippocampus. So, in the rodent brain, this is how the hippocampus is represented. So, the reason the hippocampus is most interesting is multifold. So all the brain, the concept that brain is plastic is, I suppose, everybody here understands, because even though there is this general idea that adult neurons do not divide or differentiate, they are pretty much fixed. But the connections between neurons can undergo changes. The circuitry is plastic. So hence we say that the brain is plastic; it adapts to changes. But within the hippocampus, there is a region called as the dentate gyrus. So this is how, if we take sections of the brain, the the region marked in red, that's the dentate gyrus. So within the dentate gyrus, a very special mechanism happens, something that's called as adult neurogenesis. So uh, the idea that in the adult brain no new neurons are formed, it pretty much went away with the discovery of adult neurogenesis. So this adds another layer of plasticity to this particular brain region. So you can imagine that new neurons are actually being formed all through life, and they actually get functionally integrated into the circuitry of the hippocampus. And the reason this is even more interesting is because, in response to stress, several studies have found, both from post-mortem brain samples and from rodent studies, that there is a clear effect on neurogenesis. And also, whenever neurogenesis is affected, there are implications for the memory, cognition, but also the behavior of the animals. So, hence, this region is pretty interesting. So, looking at the major symptoms of any mood disorder, uh, the uh, the the conditions that are affected are reward or pleasure sensing. So, whenever we do something, an activity that we enjoy, maybe talking to friends or eating something that we like, we feel good. So there are certain brain regions like the accumbens and the VTA and the cortex, which are involved in this response. So this is technically called as hedonia. Closely associated with this is motivation. So we feel motivated to do things because we expect a reward at the end of it. Right? What happens in certain mood disorders like depression and a part of bipolar is a loss of this reward, something that's technically called as anhedonia, the loss of pleasure sensing. So you can imagine that if an individual is unable to feel reward, they will no longer be motivated to do things. That is a major hallmark of depression and bipolar disorder. Further, another aspect that is oftentimes affected is memory and cognition due to effects on the prefrontal cortex and the hippocampus. So, so you can understand that there are several factors involved in ultimately leading to a mood disorders, right? You have stress, which affects, which leads to epigenetic modifications in several brain regions, uh, namely these, and ultimately has implications for the pathology of mood disorders. So what I and others in my lab are studying is to understand how exactly these epigenetic modifications lead to these changes. What are the particular factors or genes involved within the whole system? So different people in the lab are studying different aspects. I, in particular, study histone modifications and the enzymes involved in that. So I'm not going into the details of what data I have. Maybe sometime later, if I'm able to give another journal club talk. Uh, so, uh, so for doing this, to be able to do this, we need to have access to the brain tissue, because we literally have to probe into the brain cells and see what epigenetic changes are happening. So to make that happen, we use mouse models. So there are several animal models that I use to understand mood disorders, and most of these models are based on the association between stress and mood disorders. So the basic tenet is you stress the animals and then you score their behavior to see whether or not they've developed conditions that mimic mood disorders. So particularly in mice, this particular strain of mouse is what we use. It's called the C57BL6. There are several reasons that we use this mouse model. These are docile animals, not very aggressive, so they're conducive for behavior analysis. So, to establish a model for mood disorder, there are several stress paradigms which are standardized. So, some of the major ones that I've listed here, there is one that's called as the prenatal stress. So, it's called prenatal stress because here uh, pregnant mice are stressed. So the way the stress is given varies, but the idea is pregnant mice are stressed, and then the effect of this stress is analyzed in the offsprings that are born. So this you can, in the human condition, if you have to compare this, you can say that a pregnant mother. So we all know that pregnant mothers are advised not to take too much stress and not to get involved in heavy or stressful activity, primarily due to this. If the pregnant mother perceives stress, it has effects on the offspring. right? Then there's something called as the early life or maternal stress, where once the mouse offsprings are born, they're either separated from their mother, or some level of stress is induced so that their maternal care, that in the normal maternal care that's supposed to happen, doesn't happen. That also has implications for how these offsprings will develop, and they will have some epigenetic dysregulation, which makes them more susceptible to developing mood disorders. So, in the human condition, this can be equated with someone having a troubled childhood. So, these two models are based on early life and changes or things that happen in young age. Or and further, in adult animals, there are two major stress models that I used. One that's called as unpredictable chronic mild stress, or UCMS. So, in my lingo, I call this stress life, because that's pretty much what we go through every day. We face a lot of unpredictable, mild stressors, which, and most of us find a way to deal with them. But sometimes, if the stressors tend to be for a long time in a short period of time, that can precipitate the development of a certain mood disorders or certain symptoms of mood disorders. So that is what this is something that we mimic in animals also. But of all of these, one of the most robust stress models that exists is the chronic social defeat stress. So, to compare this to the human condition, you can say it's something like bullying. So, in this, we also use another mouse strain that's called as CD1. It's the same species, but a different strain. So, this particular strain, it tends to be inherently aggressive. So the way this protocol, so this is the protocol that I use in my study. The way this works is, we have our experimental mouse and we have the aggressor mouse. So, so we use slightly older aggressor mice because they tend to be even more aggressive. We house them in isolation before we start our paradigm. So when we house them in isolation, initially they will themselves undergo some level of isolation stress, but over time they become extremely possessive of their cage. After which, if an intruder is introduced into their cage, they will not tolerate that. They will try to chase away the intruder, try to fight with it. So this is how the stress paradigm works. We put our experimental animal in the chamber of the aggressor for 5 minutes, during which the aggressor will chase it, fight with it. So that's extremely stressful for the experimental animal. And after this 5 minute of interaction, we put this animal uh, in the same cage but with a separator in between. So, how this works is, the animals will not be able to interact with each other after the 5 minute physical interaction, but they can still sense each other. So, for the experimental animal, this is extremely stressful in the sense that 5 minutes it's being hit by this individual, and then it has to practically live with it. So, this 5 minute of interaction every day is done for 10 days. In the control group, we use a similar housing system, but two male mice are two C57 mice are housed together, and they are also made to interact for 5 minutes every day. But in this case, it's more of a neutral interaction. So at the end of these 10 days, we assess the behavior of the animals to understand whether or not they have developed conditions like anxiety and depression. So, what is another good thing or robust advantage of this model is that, despite being given such a heavy kind of stressor, not all the animals develop conditions like depression at the end of it. They all unanimously develop anxiety. That is something that all researchers have observed. But not all of them develop depression like conditions. So, like for example, if 10 animals are given this stress, only 6 or 7 of them will develop the condition. So, these 3 or 4 animals that don't develop depression like condition, they are called as resilient. So, the biology of resilience is also something that interests the field, and that is also something that people are studying. And in fact, there are a lot of indications that resilient animals have uh, slightly different regulation in their epigenetics as compared to the susceptible animals. So, how we score the behavior of these animals is, so as I've been talking, you might have noticed that I am not calling these conditions as anxiety and depression. I'm calling them anxiety-like and depression-like. because anxiety, depression, or bipolar disorder are human disorders. And in animal models, be it any animal model, it's not possible to recapitulate all the aspects of the human disease in animals. So what we see are some of the hallmark symptoms of these disorders in animals. So based on this, we score animal behavior. So to look at anxiety-like condition, there are several behavior tests, but one of the most well-used tests is something that's called as the elevated plus maze test. So in here, we use a maze that looks like this. So it's four-armed. It has two closed arms and two open arms, right? And we release the animal in either in the center or maybe in one of the open arms. So, an animal that has never undergone stress and is in its native state, even though it will also tend to spend more time in the closed arms because it's safer, it will still be curious enough to venture into the open arms to some extent. So, this is how the track profile of an unstressed animal or a control animal looks like. But an animal that has received chronic stress, it will be very apprehensive to venture into the open arms for the fear of falling off. So we look at this difference in behavior and we score whether or not our animals have developed an anxiety-like condition. To look at depression like phenotype, we check two major things. And these two major things are based on what is observed in the human condition of depression also. One is called as social interaction. So this is something that has been observed in humans too, that depressed individuals don't want to interact with others. They want to stay by themselves. So this is observed in animals too. So an animal so for this we use a paradigm in which you have this chamber within which there is a smaller chamber. So in this smaller chamber we keep an animal that our experimental animals have never seen before. So it is a novel individual for them. And then when we introduce an experimental animal into this chamber, so unstressed or control animals will spend more time around this small chamber because they are curious. They want to know who this new individual is and they want to interact. But animals which have undergone stress, they might approach this chamber once, realize that there is another individual inside, and then simply lose interest because just no reward perception due to social interaction, they are not interested in interacting with a new individual. So this is a classic hallmark of depression-like conditions. Another thing that we look at, as I mentioned earlier, also that's anhedonia or the loss of pleasure sensing. So for this, we use a test called as the sucrose preference test. So mice, like humans, also tend, like most humans rather, also tend to like sweet tasting substances. So if we train the animals to drink sucrose water, over time they develop a preference for it. They will go for sucrose water versus regular water. But once the animals start to develop a depression like condition, they no longer will care what they are drinking because they no longer can feel the pleasure out of the sucrose water. So, this decrease in sucrose preference is another thing through which we score the animal's behavior. So, at the end of all the stress paradigm and the behavior test, what we end up with is a bunch of animals that show us the behavioral manifestations of mood disorders, and the further steps are to probe into their brain and understand what changes are happening. There are several things that we do. We also manipulate the levels of these genes in the brain and then see how the behavior changes. So that is an advantage of working with live models, that we can actually see the manifestation of a gene modulation in the level of behavior. So, to summarize what I've been talking so far, we have psychological stress, which leads to epigenetic changes, which ultimately has implications on neuroplasticity and neurogenesis, which leads to modulation of neural circuits, ultimately precipitating neuropsychiatric diseases. So, this is the overview of what my lab studies and what I study. So here I'm going to shift gears and go into something that's called as the mental illness spectrum. So the reason I want to talk about mental illness, so this is more in the context of humans. The reason I want to do this is because I believe that a discussion on any disease is incomplete without addressing the clinical scenario. And this is something that I've noted, especially in Indian academia and all over, that there is some level of disconnect between the research community and clinicians. Researchers are doing their thing, clinicians are doing their thing, but do the two groups ever talk to each other? Because a lot of new things are being found on a regular basis that have implications for diagnosis and therapy and treatment. But are those things being implemented in the clinic? It's not always a one-to-one street. So I think in places like these, to make these things more robust, I think a group like Encephalon is very well equipped, because this is literally a group that has researchers and medicos in one platform. And we're all young researchers, we're all just starting out. So I think this is something that we need to start talking about from now on, right? Before I go into the detail of the illness spectrum, I'd like to talk about the difference between mental health and mental illness. So, oftentimes, people use the two things interchangeably. It's like a linear axis. A person either has they have a mental illness. That's a very general perception, which is wrong. Mental health and mental illness fall on two separate axes. So I think one of the best models that I have come across for this is something that's called as the Keys model of mental health. This was proposed by an American psychologist called Corey Keyes, and this is how this model looks. So mental health is on one axis, mental illness is on another axis. And so there are four possibilities. An individual can have high mental health with no mental illness, right? They can have poor mental health without a mental illness. And this is something I suppose is important to understand, that if someone has poor mental health, it doesn't necessarily mean that they have a mental illness. Maybe it's just it might be temporary, and it might the problems might go away with proper help and therapy. It's possible that someone has poor mental health with mental illness. This, I think, is something that everybody can understand. uh, it's also possible to have good mental health with mental illness. So this sounds counterintuitive, but um, for example, an individual who might have autism or any one of the autism spectrum disorders. So that person has a mental illness, a diagnosed mental illness, but they might be in great mental health. They might be getting the right help, doing taking the right medicine if needed, and they might be doing great and having a fulfilling life. So all of these four possibilities exist, and they are different. So this is something that we should keep in mind in the further discussion. So going ahead, now I'll talk directly about what happens in the clinic, right? How mental illnesses are diagnosed. So some of you will already know this. So the something that's called as the Bible of psychiatry. This book called as the Diagnostics and Statistical Manual of Mental Disorders, or DSM. So the first and so there are five editions of this book. The first and the second editions came about in 1952 and 1968. So these two editions were based on Sigmund Freud's idea of psychology. So Sigmund Freud is popularly known as the father of psychology. So so these editions were based on his ideas. So what he said, that mental illness is the result of a conflict of internal drives. So I won't go into the details of this because it's a little complicated and also controversial. Uh, as of today, most of Sigmund Freud's ideas are not really followed in the practice of psychology or psychiatry. Then after this, the third edition came about in 1980. This is when a bunch of psychiatrists realized that people with same symptoms are actually being diagnosed as having different disorders because of the controversies of Freud's ideas, and so they shunned that approach, and they went with Emil Kraplin's idea. So what Kraplin said was, so Kraplin was primarily working on schizophrenia and bipolar disorder. So at that time, during the late 70s and early 80s, there was some confusion on the distinction between schizophrenia and bipolar disorder because the two diseases have certain symptoms that overlap. But what Kraplin said is that the two diseases are distinct and he put them in separate categories. So, what he proposed was a category approach, which was then adapted to all of the mental illnesses, and that's how the third edition and also the fourth edition, which came about in '94, both of them are based on the category approach, where illnesses have been put into different groups. And then in 2013, the latest edition came up, that was DSM-5. But lately, there has been this increasing idea of going away from the category approach and adapting something that's called as the dimensionality approach. This was started by David Kupfer, who's a psychiatrist, and other people. So, what is the dimensionality approach and why is it required? So, this is primarily based on the idea that clinicians in practice have realized that an individual with mental illness does not always fall very well into one category. So statistics say that one fifth of people who fulfill the criteria for one DSM-4 disorder meet the criteria for at least two more. So classically, these things have been called comorbidities, like anxiety and depression. A lot of times occur together in individuals, and this has been called as a comorbidity. So, but this is observed so often that psychiatrists struggle to diagnose an individual and put them in the category of a particular illness. So what happens is that an individual with a mental illness might get diagnosed with one disease when they go to one psychiatrist and another disease when they go to another psychiatrist, and this can be very distressing and in the long run quite counterintuitive to therapy and treatment. Further support for the idea that it's difficult to categorize mental illness is comes from the study of genetics. So in this thing was done, I think, in 2009 or 10. So this Jordan Smoller's group from Massachusetts. So they did a genome screen from 33,000 patients. It, having five different disorders. And what they found is that there are only four chromosomal sites that are associated with all the five disorders of autism, ADHD, bipolar, major depression, and schizophrenia. What this means is that there are similar mechanisms or similar pathways that are affected in all of these different disorders, which further says that it's difficult to categorize them and the symptoms are going to overlap. So, what this dimensionality approach primarily says is that instead of looking at behavioral symptoms and putting individuals in different category of mental illnesses, what we need to adapt is the spectrum approach and this idea that mental illnesses lie on a spectrum and the diagnosis will be based not on symptoms but on measurable quantities. So an example of this, one of the spectras that have been proposed, so this one is called added dimensions. So this was proposed in 2010 by Michael Owen's group. So what this spectrum has done is put mental retardation, autism, schizophrenia, schizoaffective disorder, and bipolar and unipolar disorder in a single spectrum. And so, so this is the clinical syndrome. These are the symptoms, and you can see that three of these disorders are known to have cognitive impairment, and also several other symptoms overlap. So instead of putting an individual in a group and saying that the individual either has autism or schizophrenia, you can say that the individual has these symptoms based on which they lie on a particular point on this spectrum. And then the person's therapy and treatment will be based on wherever they lie on the spectrum. So before the DSM-5 came up, this group and also David Kupfer and all these people were trying their level best to get this category approach to remove and the spectrum approach being adapted, but it did not happen. because several people in the field that the research is pretty premature and we can't make the transition as yet. And it's difficult to say which of the two schools of thought are correct. And as David Kapoor rightly said, that the plane is already in air and they've had to make changes while it is still flying. So here again, the transition or changes that need to happen in the clinical scenario are not easy because things are set in their ways, and research is very dynamic. New things are being found every day, but its translation into clinic is something that we struggle with. So in this context too, that has been happening. So what I want to so I'm pretty much done with my presentation. The idea that I want to leave everyone with is that as young researchers, is there a way that we can make this better? Is there a way that we can have better interactions between the research community and the clinical community? And changes that are happening in the clinic, changes that are happening in research, how do the two should ideally be going in parallel? How do we make that happen? And also, what does this mean? Let's say that the spectrum approach is adapted. The whole diagnosis of mental illnesses gets U-hauled. What does that mean for research on mood disorders and other psychiatric disorders? Because we've been using animal models which do not recapitulate all of the symptoms of these diseases. But should researchers also be restricting ourselves to saying that I only study depression or I only study bipolar disorder? Or rather, we should also be looking more at which factor affects which particular behavior or which particular symptom instead of talking in variable terms. So these are the questions that I want to leave everyone with, and that's all. Thank you, and I can take questions if there are any. I hope there are. So yeah.
Yeah, there are two questions in the chat, so
Mhm.
I'll let Luke ask
Yeah.
his. He hasn't replied. So, Madhuri says hi. When the young mice was introduced to the old CD1 aggressor for 5 minutes every day, did the young one experience some hostility every day, or
Mm.
was there a difference in every consecutive interaction?
Okay. So here, the idea is that if we let the CD1 or the aggressor interact with the same C57 every day, over time it does get habituated, and its level of aggression might go down. So what we do is we follow a rotation system. Every day the C57 gets introduced to a new CD1. So for the CD1, this animal is again a new animal. So we ensure that there is no great variation in the aggression that is observed.
Okay, I have one question related to this. So, in that test, how quickly were the effects of depression or anxiety manifested in the mice? Just after the 10 days?
Yeah, so we do the behavior test typically within 24 hours of the last stress episode, and most times, if the paradigm was done correctly, we do see the symptoms.
Luke has a question. So he asks, are there any measures of knowing the extent of neurogenesis, like levels of BDNF or something?
Yes. Not the levels of BDNF itself, but there are several markers for neurogenesis. So some of these markers, like SOX2, that people might have heard in the context of stem cells. So SOX2 is also expressed in neural stem cells. There are other markers called, there's something called as double cortin, that is abbreviated to DCX. So that's found in newly differentiated neurons. There is also a protein called as nestin. So nestin is it's a part of it's a type of intermediary filament that is found only in neural stem cells and neural proliferating cells. So there are several markers. So in fact, if you just look at the neurogenesis literature, you'll see that most of the studies have done either molecular studies or to measure the level of these proteins, or done IHC to visualize these proteins. So yeah, it's possible to know how neurogenesis is going down when it's going up. The difficulty comes in understanding the fine tuning of neurogenesis. So you can, because if you have to look at the animal brain, You give the stress, then you sacrifice the animal, make sections, and observe. But how exactly do the changes happen over time? Because some studies, like there are very preliminary studies which say that there are changes within a day. How do you capture this? Because once you sacrifice the animal, that's it. How do you capture the plasticity of it is the challenge.
Okay, so there's one question. So Aditya asks if you could explain the plus test once more. He didn't get it.
Yeah, I'll go back to it. Just a second. Okay. In the plasmid test, so there are four arms, right? There are two open arms and two closed arms, right? And this maze is slightly elevated about 100 centimeter from the ground. So basically, if the animal is on the open arm, there is a chance that it can fall off. So animals which are unstressed, control animals, even though they know that there is a chance of falling off, they are still curious enough to venture into the open arms to some extent. But animals that have been stressed, their risk taking behavior is not as much. So they will barely venture into the open arms, and this is the difference that we look for. I hope that makes it a little clearer.
Okay, so Daniela says, "Hi, nice presentation. Have you considered the role of low inflammatory response developed by cortisol itself in the epigenetic changes?" To so first you answer this one, then we'll go to the next.
Yeah. So inflammatory response in response to cortisol, yes. So the inflammatory response doesn't lead to epigenetic changes. What the idea is that epigenetic changes lead to all of these changes, right? So stress directly affects epigenetics, which further affects the production of cortisol, inflammatory responses, and several other things. So that is the general idea. And in fact, there is enough literature to show that epigenetic changes directly have implications on the functioning of the HPA axis and other inflammatory neuroinflammation and other things.
And the second part of the question says, have you found epigenetic changes in the region that codifies genes located to the mitochondria?
The
This is
answer is
because
yes.
mitochondria. Yeah.
Yeah. Mitochondria respond to molecular stress also. And the answer is yes. So there have not been like direct correlations between a particular psychological stress leading to changes directly in a mitochondrial gene. But when we look at epigenetic enzymes that have implications for epigenetics, a lot of times their targets are mitochondrial genes. Like, for example, in my lab itself, we have found some preliminary studies that a particular demethylase, which gets modulated on stress, some of its targets are mitochondrial genes. In fact, like that is something that we've been working on, and hopefully the paper will be out soon. So, yes.
Okay, so Kirtana asks, are these epigenetic modifications also seen in glia? What role does glia play in neuropsychiatric disorders?
Hmm. Okay. So epigenetic modifications are also seen in glia, and so the role that glia play in neuropsychiatric disorders, like there is no simple answer to this. They definitely do play a role because glia are responsible for maintaining the correct environment for neurons, and also the levels of myelin and the levels of neuroinflammation. So they do have very important functions, and changes in glia also have implications for neuropsychiatric disorders. In fact, this is something that is being done in the field. There are people who study specifically the effect of or the effect of stress on glia and further on neural circuits and behavioral manifestations. Important in this would be something that's called a single cell transcriptomics. Like, you give the animal stress, you take whichever brain region that interests you, and you separate neurons in glia. And using single cell transcriptomics, it's possible to find out what exactly single cell transcriptomics and other high throughput studies in individual populations to understand how one kind of stress affects the two populations differently. So yeah.
Um, so Pradeep asks, do you guys test effect of different ages in mice in which you
So
do those tests?
we have not done that in the context of mood disorders, but yes, there are several other groups which do it. They compare juvenile mice versus adult mice versus old mice. In this context, yeah, there was one study in my lab that did involve some aspect of studying the effect of age. So that study was based on vitamin deficiency. So we were looking at how vitamin B deficiency has implications for stress response and behavior. So there, adult mice and old mice were compared, and the effects were more pronounced in old mice. So yeah, age definitely has some effect on behavior, and it's possible to capture that in animal studies. Yes.
He also asks, isn't it possible to study the effects of stress on neurogenesis by using real-time imaging like two-photon calcium imaging?
Yes, it's possible to do that. The only again people have tried to do this, and there has been some success, but then again, it's not as the what do you call it? The resolution is not as good as what one would hope. But yes, this is possible to do. In fact. I was recently reading a paper which I can send that paper over to the group. Yes. Yeah.
Great. Is that because of these regions lying deep
Mhm.
inside the brain,
Yes.
so
Yes.
less accessible?
Yeah. So these are all deep brain, especially the hippocampus. It's pretty much at the like it's a few
Bottom part, yeah.
centimeters inside. So yeah, it's difficult. Even when we have to do like electrophysiology recordings in these brain regions, it's very
Yeah.
difficult because the electrode in vivo recordings, that is, it's yeah. So these are practical problems. Yes.
Okay, Kirtana says, speaking of age, she was wondering, are there any differences in stress responses between male and female mice?
Oh yeah, I'm happy someone brought this up. Okay, so the whole field of neuroscience in general and neuropsychiatric diseases in particular has been focused on male animals, right? And the reason being, especially for behavior studies, because female animals like humans do undergo something similar to the menstrual cycle. That's it's called the estrous cycle in them, which complicates their behavior, and so the scoring of results is not so easy. It complicates data analysis. So for the longest time, people have been ignoring female animals, and I think in the past decade or so, this also has been picked up. And yes, differences in stress response have indeed been shown in males and females. And there are studies which have shown that there are distinct pathways that get regulated in the two animals, even though the behavioral manifestations are similar. So yes, and so this has great implications again for clinical translation, because it basically means that you might have to give different medications to men, women, and also another complication in the human condition is gender is not just restricted to just male and female. So yeah.
Mm, nothing else. What was? Okay. So Priyanshu asks, okay, yeah, go
Yes,
on.
yes. Mm-hmm. 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 Okay. So this is a field that even I don't know a lot about, but I would say that it's difficult because estrous cycle essentially is different from menstrual cycle. So menstrual cycle is periodic; it happens once a month, typically. But estrous cycle is not like that. It is not as periodic, and it goes on for longer. So direct correlation would be difficult, but it's possible. And people have tried to do some studies based on that, only recently, that is. But I am not sure what the results are. But it's an interesting point, and I think it's worth pursuing this.
Yeah, just to add on to that,
Mhm.
Kirtana, your question about differences in stress responses between male and female mice. So there was a study which we were trying to figure out the differences in adapting to stress in case of MSEW mice, which is maternally separated and early binding mice. So we were seeing this disparity in male and female mice. So that lab mainly worked in on olfaction, and there
Mhm.
is a lot of literature which claims that because of the stress, there might be maladaptive responses in the olfaction sensing mechanisms, because it's also based on the reward sensing mechanisms.
Right.
So in
Mm.
that, we actually observed that the female mice were better as compared to the male mice when it comes to handling stress, if you go by this criteria. But again,
Mhm.
there are also concerns about larger sampling sizes. So that study is being done with larger sampling sizes now as well, if I'm aware of it.
That's interesting.
Yeah,
So,
and sorry,
yeah, yeah, continue, please.
no, no, go ahead, go ahead.
Yeah, so this reminds me that so there was a study from one of our collaborating labs recently that was on hypoxic stress, though. So it's not a psychological stress, but it's like a physical stressor, hypoxia. And the study was in zebra fishes, and there also sex-specific differences were found, and they also found that so the effect of hypoxic stress, it becomes so females respond faster, so the damage that is found in females is higher sooner, but compensatory mechanisms also function better in females. So yes, there are definitely sex specific differences in several of these contexts, and yeah, it will be very interesting to look at those in the context of adult stress induced neuropsychiatric disorders. Yeah,
Yeah,
I think that
yeah.
is something that
If
yeah.
I could just use this opportunity to ask my own question regarding
Mm.
this, JC. So my question was, if there is any correlation which you might have observed or which you are going to probably study between the reward sensing neurotransmitters such as dopamine and serotonin and the epigenetic modifications. So, for example, maybe in dopamine deficient conditions, there would be some epigenetic modifications which are dysregulated, or vice versa. Or is that a correlation? Is that a cause effect relationship? I mean, has there been any studies on the relation between these two?
There have been studies on the relation between these two. I can't quote specific examples, but yes, I have read studies that where which have found that I think there was a study that was on DNA methylation, acute stress leading to changes in the methylation status of some of the dopaminergic receptor genes, which has implications for dopaminergic signaling. So, like my study doesn't involve that angle. We look more at neuroplasticity and neurogenesis, but yes, there are other groups which are looking into that, and there is literature. Yes.
Okay, thank you.
So, Shresh asked this question. Is there any other question? Okay, so
I'm not sure if we addressed this
Priyansh, you
question.
asked.
Yeah.
It was crisp. It was crisp and great presentation, especially for me who is new to the field of neurobiology. He wanted to ask a question, which is, when we say stress, what does the physical basis of it mean?
Hmm. Okay. So yeah, cellular stress, like we talk about in oxidative stress, that is one of the. So yeah, this is an interesting question, and the answer is we don't know. We don't really understand how exactly this stress directly affect epigenetic changes. So initially, people were not understanding how stress changes behavior. That people figured out key, okay, it could be epigenetics. But how is stress leading to epigenetic changes? What is the physical basis of it? We don't entirely understand. Oxidative stress is a candidate that's being considered, because but then again, oxidative stress comes about when your brain cells are overworked, when there is an then there is a dysregulation in brain energy metabolism. But how exactly that's leading to epigenetic changes is not understood. Yeah.
So, how do epigenetic changes occur? I mean, what triggers epigenetic changes to occur?
So what we look at is a dysregulation in the levels of epigenetic modulators, right? There are enzymes and proteins that need to be expressed to a certain level to maintain a certain you know uh, map of epigenetic, right? So when there are changes in the expression of these proteins, you see changes. It gets translated into the differences in these modifications and ultimately affects the expression of other genes. But in the first place, how are these genes getting modulated? Like the question that Priyanshu asked, that there is a stress, that like you perceive stress, but how is that changing the levels of some of your genes? What is the physical basis of it? We don't entirely understand that.
So, what I was asking was, do we know some of the biomarkers that might be related to inducing that epigenetic change?
So yeah, one candidate, as I mentioned, oxidative stress is one. Another thing that people have mentioned is like the glucocorticoid receptors and how cortisol binds to it. So when there is too much cortisol and there is too much activation of glucocorticoid receptors, this seems to have implications for the expression of some of these epigenetic modulating genes, which further leads to epigenetic changes. So that is one aspect. But still, it doesn't explain the entire range of changes that we observe.
Okay, one more question related to that is, how is this window of epigenetic changes happening at a more rate in, say, in the early life, or
Mm.
does it carry on like till late life? Is it the same?
Hmm. So in early life, I wouldn't say the rate of epigenetic change itself has been found to be different in young animals or older animals, but in early life, the plasticity of the brain is higher. So epigenetic changes have a potential to have greater impact on a younger brain as compared to an adult or an older brain, right? But there is no difference in the rate of epigenetic change itself. The implications will be different. Yes.
Okay, so Kirtana has a question. He says, this might be a stretch, but are there studies that look at exposure to stress and cognitive decline in later stages of life, possible linking to dementia,
Oh
maybe?
yeah, yeah. Oh yes. So people are studying that, like in models, in dementia models or in neurodegenerative disease models like Alzheimer's. People have tried giving stressors to these animal models and seeing whether it precipitates or makes the neurodegeneration worse, and it has been done. As you might expect, the answer is yes. In fact, one of my colleagues is actually working on a similar project. So their model is ALS, or amyotrophic lateral sclerosis. So they have a knockout animal, and the protein knockout, the particular protein or gene, has been associated with functions in motor neuron regulation. So their hypothesis is that the knockout animals should show some of the symptoms of ALS. But in the baseline or in the animals, they've not been able to show any gross morphological or motor changes. So what they're trying now is to stress the animals and see if some of these symptoms come up.
Okay.
So yeah,
So would that also be due to something like humans and rats are not similar?
definitely. There, you since we talk of homologs, but there are several genes that are present in humans. They're not present in rodents. Several genes present in rodents, but not present in humans. So yes, a direct correlation is might not always be possible. So this is also something that. So hence, when people say that we are studying animal models, it's also important to study what animals have that humans don't have, because some of the translations that might have already been made might not be correct. So, yes.
Okay, so we don't have any more questions, as I see. Okay, mm, yeah. So we have another question.
Mhm.
Uh, on, yeah, sorry, I
Oh,
missed it.
yeah.
On such differences in murine studies, I was curious about
Yes.
adolescent male mice and andropause. Does there change in
Mm-hmm.
response?
Mm-hmm. Mm-hmm. Oh, I honestly don't know much about this. And I don't know if there are any studies in this context. I'll have to look into it. I assume there might be, because people have studied differences in stress response in adolescent versus adult animals, and also juvenile versus adult animals. But specifically looking at the phenomenon of andropause, I'm not sure if that has been done.
So that's it. I guess we have exhausted all our questions.
Yeah.
It was an amazing talk. Thank you.
Thanks a lot. Yeah. And so, if any of you have any ideas or want to soundboard or brainstorm, you can mail me or just DM me on Twitter, whatever works. I should have mentioned my mail in the
Annapurna,
presentation.
regarding that, could you probably enter your email ID in the chat
Sure,
so that
sure.
people could reach out?
Yeah, I should have put that in the presentation. Forgot to do that. And my Twitter handle is, so thanks a lot, Encephalon, for making this happen. Yes, I enjoyed this.
We did too. At least I did. That's for sure.
Yeah, thank you, thank you. Yeah, and also I think this the discussion between research community and clinical community. I think that is something that you all are very well placed to take up and
Yeah,
should consider that.
I agree to that. We could probably pitch in that idea to the group, and we could have more engaging discussions between the clinical and the research community.
Yes, yes,
It is tough though, infrastructure wise, our institutions in India aren't quite developed in such a way that you could collaborate with medical students. Even medical doctors aren't very enthusiastic about research. So,
yeah. There are going to be hurdles in this, but I think we have a new generation of doctors coming up. We have
yeah.
people like Pranjal, we have other
Yeah,
medicos in the group. So I think the future is bright, and it's possible. Like I think one field that has been doing pretty well in this is the group that studies muscle degeneration. So a lot of conferences have been happening where they're getting together researchers who study muscle development and degeneration, and patient groups for muscular dystrophies, and clinicians, and having like a entire discussion on what is required for each group and how can all groups work together to actually make things faster or more meaningful. So yeah. Mm. Mm. Sure. Thank you. Thanks for having me.
Also about Neuronov, we are having our
Mm.
registrations open, so you could
Mm.
check it out and check out the delegates, not the delegates, the speakers. We have
Speakers.
a great lineup,
I
at least
I don't
until
doubt
now.
that. Yeah, I'll definitely register for this. Yeah.
And there are also some competitions that people
Mm.
can take part in, so feel free to check it out and drop in your registration.
Thank you.
So
thank you so much.
Yeah, thanks a lot.
thank you so much, everyone. We'll
Bye.
end this meeting now. Goodbye. Have a
Yeah.
good night or good day wherever you are.